The views expressed on Science for the People are not necessarily the views of this station, its affiliates, sponsors, or advertisers.
This week on Science for the People, we're looking at how people and pop culture understand the ever-evolving science of the brain. Hello and welcome to Science for the People. I'm Rochelle Saunders. My first guest is Dr. Cliodhna O'Connor. She's a postdoctoral researcher in psychology and language sciences at University College London. Her research focuses on public engagement with science, and she was a co-investigator on a grant awarded by the Faraday Institute of St. Edmund's College, Cambridge, which explores the popularization of neuroscientific knowledge and its implications for social life and common sense beliefs. I spoke with Cliodhna at a conference I attended in Cambridge this fall, put on by the Faraday Institute, which looked at the uses and misuses of biology in the media. So thanks for speaking with me today.
No problem.
When we talk about science, we so often forget what happens to it when it enters a more mainstream audience, and it mixes and intermingles with our existing culture, worldviews and ideology. And your papers really zone in on this topic. Was that intentional, I'm assuming?
Oh, yeah, definitely. I mean, a lot of theorists have argued that in contemporary society, science is actually one of the key influences on our common sense knowledge. So whereas previous societies in other historical eras might have relied maybe on religion or the wisdom of elders, we increasingly turn to science to understand the world around us at a very day-to-day level. So I'm a social psychologist, so I'm very interested in mapping the very kind of nuanced processes by which scientific information enters society, how it might be transformed along the way, and how different people with different interests, different agendas, different backgrounds might read different things into that information and then use it for different social purposes.
You mentioned in one of the papers that there's currently very little research exploring sort of neuroscience's public image, which I actually found really surprising. Why do you think that there hasn't been a lot done on this topic?
It's definitely accumulating at quite a rapid rate. I mean, I think, so when I wrote the paper, that was probably 2 or 3 years ago. So at the time, it was still very much at the start. I mean, the public prominence of neuroscience today is, I suppose, it's been gradually accumulating since the 90s. So it's a relatively recent development. So I suppose social scientific research has taken a while to kind of respond to that and to incorporate that into its own analysis. Another issue is that analysis of this so far has been largely based around maybe sociologists or cultural commentators or people in the humanities who've looked at this in quite a conceptual way, who've just kind of looked at what's happening around them and given their opinions about what's happening or applied their own theoretical frameworks to look at what's happening. But there's been very little systematic research that has tried to empirically document how exactly this is happening, in what context it's happening, what effects is it having. So rather than imposing a kind of predetermined theoretical framework on it, to try and gather some data first and then use that to develop a more conceptual analysis of what's going on. So I think that's really starting to get off the ground now, and we're seeing more and more people, there's more and more data being generated, and it's converging on some pretty interesting results.
Interesting. So it sounds like there's been a lot of conversation, but not a lot of data gathering.
Exactly.
So your papers focus in on neuroscience specifically, and I'm wondering, do you think that neuroscience, the way neuroscience is currently being interpreted in the media, is problematic? And is it more problematic than the way other sciences are being interpreted?
It's difficult to make a universal statement about whether it's problematic because it's interpreted in so many different ways. So there are definitely problematic elements to it. There are issues with it being used to reinforce particular elements of social hierarchy, particular stereotypes about a wide range of social groups. At the same time, Other people have used it to different ends. So, for example, it's being in certain aspects of mental illness, it's being used in quite a positive way as a kind of stigma reduction mechanism, to kind of show that this is a biological contingency that people are operating under, that they might not necessarily have an awful lot of control over this, and then that it's not their fault. At the same time, it's not necessarily, even within the mental illness field, it's not necessarily a universally positive. Research has shown that in certain contexts, very biological conceptions of mental illness have been used to distance mentally ill individuals from society, from healthy, normal society, portraying them as kind of inevitably, intrinsically wrong, as opposed to the rest of us. So very difficult to make a blanket statement about whether it's helpful or whether it's problematic. But I think what I and other people have been trying to do is to apply empirical social scientific techniques to identify the particular contexts in which it's being used in problematic ways and the particular contexts in which it's being used in more helpful ways.
I'm curious as to how much of this interpretation, leaving aside positive or negative interpretation and its ultimate effect, but how much of the interpretation is being driven by the media as opposed to how much of it might be driven by the scientists who are working on the research, or potentially the public relations departments that are creating the press releases. There seems to be so many different places where you can get these layers of interpretation before it gets to a member of the public.
That's definitely the case. There's a lot of actors involved who have different parameters that they're working under. They have different roles to fill, and all those things impact on how they construe and transform the information that they're working with. But I think it's important to maintain sensitivity to that complexity rather than trying to separate it out and create quite a simplistic sense of it's the media's fault or it's the scientists' fault. I mean, the language of fault and blame probably isn't that helpful in all this anyway. A lot of this is quite natural social communication processes. It's not necessarily something that someone should be really condemned for, but it is maybe something that we should be sensitive towards. So, I mean, I think all of the agents that you mentioned have an involvement in this. I mean, scientists choose to study things in a certain way. Many scientists would say that that's a completely objective, neutral situation, but it's not. You know, you choose to study certain things and not to study other things, and that's often driven by funding, by government policy. What support is there that is funneling scientific activity down certain directions and away from other directions? In terms of public communications, my research definitely suggests that the press release is one particular point at which a lot of the transformation of knowledge happens. It's often journalists' only source of information about the research. Journalists are working under very heavy time constraints, and often they can't actually access the original scientific paper because it will have been published in a journal that you need to pay hundreds of pounds to actually look at in the first place. So there's an issue of access there too. But yeah, the press release definitely sets the tone for a lot of the media coverage that develops. And then journalists will insert their own agendas, their own interests, and those of their readers into the information that they're conveying. And then readers will interpret that in very diverse ways as well. So it's a very kind of constantly shifting field. It's a moving target.
It's really fascinating because it seems like the level where the interpretation happens, at least in a field like neuroscience, is where people begin to speculate as to what does this mean, or how does this apply to my life. Mm-hmm. So to some extent, it seems like perhaps the absence of labeling some of this stuff as speculation becomes somewhat problematic in the way people absorb the information.
It's definitely something that people can really kind of relate to their own personal circumstances. And that's not necessarily a negative thing. Sometimes that can be a source for bias, but also that can be a source for a very, very vital public engagement with this type of science. You know, you can produce this very dynamic context that you might not be able to get with other forms of science that might not have that immediate relevance to people. So I think it's important not to pathologize it when people do personally invest in these things, and actually to kind of think about that as a resource that we can create this very healthy public debate around the scientific knowledge that's being produced.
This is Science for the People, and I'm speaking with researcher Cliodhna O'Connor about how neuroscience is absorbed, interpreted, and often misinterpreted by the public and the media. I'm aware this hasn't been covered in your work to date, but I'm curious if either it's on a roadmap or if you've heard of other people doing research into this. Obviously, the media can't cover every press release it receives, and I'm interested to know if there's any research that's been done on what hasn't been covered as opposed to what has been covered, and the choices people make to pick this bit of research over this bit of research to popularize or publicize.
That's really interesting. I'm not aware of any research that's looked at that systematically, but definitely there are certain cultural and media trends that certain things tap into and other things don't. So I can probably speak more about what's always covered than what's never covered. So one of the things that's getting a lot of attention is dementia and ways to prevent dementia. So what are the techniques that you can adopt to work on your brain and through that maybe reduce the risk that you will develop dementia in later life. Now, a lot of people have applied a lot of critique to this strand, both because in a lot of occasions the actual claims that are made for specific brain training techniques are very overblown factually in terms of what the scientific evidence tells you is the benefit for that. And also because this kind of constant call that you should be constantly attending to yourself, working on yourself, applying discipline and monitoring in everything that you eat and everything that you do, it can produce quite a restrictive cultural environment, induce a lot of worry in people if they're not doing these things, or even guilt in people who subsequently develop the condition that they should have done more, that it's their own fault. So there's a lot of issues around putting responsibility within the individuals to within already pretty harassed individuals who have a lot of concerns, and putting yet another almost moral obligation on them to care for themselves, to ensure their own health, to not be a burden on society.
So I'm wondering, for the sake of our listeners who haven't had the opportunity to read the papers that I did, I'm wondering if you could quickly summarize some of the themes that you found, the major themes that you found throughout the process, as when it comes to neuroscience as it's represented in the media.
Yeah, sure. So one of the themes is what I've just been talking about, this idea that you constantly have to work on your brain to either prevent it from degenerating, from contracting some type of neurological disease, or just to improve yourself as a person, improve your work performance, improve your educational performance. And this has been linked to a lot of sociological work, a lot of work in Foucauldian traditions, and other traditions that have focused on how the self is put as the responsible agent for producing a society, a particular type of society that our Western individualist culture encourages. So that's definitely one major strand that's emerged in my research, looking at how, at the discourses that neuroscientific information is drawn into in the media and in other contexts. Another pattern that I've been particularly interested in as a social psychologist is how ideas about the brain are used to underline differences between categories or groups or types of people. So whether that's about the male brain versus the female brain, the gay brain, the depressed brain, the addicted brain, you have this whole spectrum of groups, of divisions that exist in our culture that are suddenly portrayed as determined by biology, by inscribed by our biological makeup, really relegating any role of history or society in cultivating the types of categories with which we're familiar with now. And the way that these categories and the projection of brain information into these categories, the way it's represented, it often is done in quite a regressive way. It often portrays these categories as completely homogeneous entities, very little sense of individual variation within the categories, and often reproduces very old, often quite destructive, stereotypical information about the individuals within those groups. So that's been another trend that I have been particularly interested in my research.
Fascinating. What I find really interesting about reading about neuroscience in the media is the stories that seem to get the flashiest headlines and the most attention are, like you say, either kind of polar opposites. This is, this half of the population is one way, this half is the other way, and this is the difference. I'm thinking sort of some of the male-female brain dynamics. And then you get the deviations from so-called normal. What I find really interesting is we very rarely hear or talk about what that normal means. And I'm interested, it's one of those sort of gaps, is what is a normal brain? And is it the same in every context?
This is a really, really important point. You have to kind of think about the visual dimension of this as well. So often, say in the media coverage, you'll see two MRI images, very kind of differently coloured, differently shaped, and they'll be placed right next to each other for the purposes of explicit comparison between them. And one will be labelled normal brain, and the other one will be labelled some other category, which is by implication abnormal. So it might be an addicted person or a person with a particular mental illness. And there's a lot of information given then about the characteristics of the abnormal category, what this neuroscientific information implies about their behaviour, their prospects for the future, their treatment. Very, very little information given about who exactly was this normal person, on what dimensions did you decide that they represented the spectrum of normality. So I think, there's definitely a lot more work to be done in decoding how normal is represented in this and what are the assumptions that have gone into that.
It's also really interesting that quite often when these comparisons are made, they do appear to have a very deterministic quality about it, as in, this is the abnormal brain, and therefore this person has done this, or this person acts in this specific way. Whereas, if you really think about it, there must be a diverse range of possible behaviors and a diverse range of effects that this may or may not have. Which I find is notably absent from many mainstream articles. Yeah, I mean,
most lay people and scientists both would accept the proposition that nature and nurture interact in the formation of individual variation in behaviour and personality and psychology. But because the neuroscientific information holds this really kind of striking rhetorical power, it's seen as very real and immediate, it often crowds out all the other types of explanation that are also equally relevant. So often there's very, very little attention given to the other influences, the environmental influences that might mean that a person who's born, or two people who are born with the same biological characteristic might be led to very, very different outcomes as a result of the experiences that they've had. So it is definitely portrayed in a very, very deterministic way in many contexts. And I think this is especially worrying when it's absorbed into public policy. So you've seen a lot of people in think tanks and other policy contexts who've speculated that with the development of scientific technology, we could reach a point where we're having routine MRI screening of newborn babies, identifying which ones of those are going to be criminals or a drain on society in some other way, and then targeting interventions towards those individuals at a very early stage in their life. Now, there's a lot of ethical problems, I think, with labeling a newborn baby as a criminal. It almost becomes a self-fulfilling prophecy in a way. Obviously, then their parents, their families, their teachers are going to start responding to them in quite a different way. And if that child grows up believing that they have that behavioural tendency, that's innate, that might actually push them towards that. Or at least just provide a very kind of emotionally unpleasant circumstance and a lot of social stigma. So the deterministic element of this has a lot of implications in a lot of different contexts, not many of which are very positive.
You're listening to Science for the People with Rochelle Saunders, and we're discussing the way the public interprets the neuroscience they read about in the news with Cliodhna O'Connor from University College London. It was really interesting as well to read the paper that you wrote that was primarily about discourse on neuroscience as it applies to parents and infant and child development. That was really fascinating, and as I'm not a parent myself, but as I was reading through the article, I couldn't help but feel so sorry for new parents getting this deluge of information. Some of it, I'm sure, not well substantiated in things like parenting books, and where people have kind of grasped onto some mention in a study somewhere or something that they read. And ultimately, some of this stuff seems to be put on parents in a position of potential blame, whereas if you do this wrong now, your child is screwed up for eternity. There's no going back after X date or X point. One cup of coffee could be the difference between this and this, which strikes me as terrifying as a new parent.
Yeah, definitely. I mean, it's a lot of pressure to put on people who are already fairly anxious and vulnerable. And I think it's also important to point out that these messages aren't directed at all parents equally. So firstly, they're overwhelmingly directed at mothers. It's assumed that mothers are the ones who are the primary caregivers and who are responsible for developing their children in the optimal way. So you see a lot of blame of mothers who work outside the home, mothers who might be on certain medications during pregnancy, basically suggesting that if their child goes down the wrong path in any sphere of life, that it was that mother's freely taken choices that were fully responsible for that. And the other way in which these discourses are differentially targeted is that they are overwhelmingly directed towards people in lower socioeconomic circumstances. So it is women who are in particular communities who are targeted as the needed recipients of this advice. Because people believe that it's certain communities that are going to give birth to and cultivate the criminals and the antisocial elements of the future. I think there needs to be a lot of attention paid to the way that these messages are differentially directed at different groups and can function as a sort of social control or excuse for intervening in some groups and not others.
It's interesting that neuroscience is often talked about generally as something that's going to revolutionize our understanding of parts of what it is to be human, or how we interact as people, or how behavior is determined. And yet, what I found your research quite often shows is that a lot of this information is being taken in and absorbed into existing biases or existing stereotypes. So it seems to be quite the contrary in a lot of cases. It seems to be reinforcing what we currently think rather than being radical or revolutionary.
It's definitely the case. And a lot of this is what, it kind of relates to what I was saying earlier about the disjunction between there having been a lot of conceptual analysis, theoretical analysis of this new phenomenon, but very little empirical analysis. And the theoretical stuff that has kind of just looked anecdotally at some evidence around them but hasn't actually systematically collected any evidence, that has tended to suggest that neuroscience is this really, really profound new source of knowledge that has very radical implications for how we think about ourselves and how we organise our society. But increasingly, as the empirical data is accumulating, we're seeing that it's actually the opposite, that people are interpreting this information through the lenses through which they view the world already, and it's actually being used to further entrench existing aspects of our social system. It's putting a shiny new gloss on old cultural biases rather than completely tackling and overturning existing ideologies or worldviews.
So is there a strategy for neuroscientists or researchers to help mitigate some of these extra layers of interpretation that are being added on that perhaps weren't intended? Are there steps that some of the researchers can take to mitigate some of this?
I think it's hard to give a step-by-step practical guide because all the different contexts are going to differ so much. But I think what's really important is that scientists become aware. We need to cultivate sensitivity to the social and ethical implications of this research. I think in a lot of scientific communities, there's a tendency to just blame the media. It's all the media's fault. We're just doing pure neutral science, and then it's distorted by the press. And I think that's a bit of a cop-out. You can present information in certain ways that definitely encourage the media to take it up in particular ways. And certainly my research has suggested that scientists can be very, very active behind the scenes in encouraging particular interpretations of their research, or equally can be completely non-active in discouraging other more problematic interpretations of their research. So I think scientists need to be aware of the spectrum of values to which their research might relate. And maybe they could, in presenting their research, specifically suggest that it might have this type of implication and specifically disown other implications that they might be cautious to avoid.
And on the flip side, I'm just thinking of new parents who are reading all of this information. From their standpoint, is there a mindset or a way that they can read some of the newer research that's coming out in a way that they don't feel blamed, or perhaps is a better way for them to do so to prevent some of the anxiety that's being associated in those cases? I'm just thinking as a layperson, mm-hmm. what are some ways you can read this stuff and not maybe overinterpret or pull it into your own worldview when perhaps that wasn't the intent?
I think people don't really pay enough attention to the importance of a relaxed environment in promoting a healthy child's development. So whether that's a pregnant mother who's kind of happy and tranquil, that will obviously be reflected in her biochemistry and will, possibly help ensure that the chemical environment in which her baby is living is a healthy one. And also in your day-to-day interactions with children, it's all very normal stuff that we know. It's having a sense of humor, having that element of just a happy, carefree environment, I think can often be much more important than a very, very structured, regulated, planned environment where everything is determined in order that it has a particular outcome. And in doing that, you lose a lot of the more spontaneous elements that can be very important in building an emotionally happy household.
Well, thank you so much for talking to me. It's really interesting research, and I look forward to seeing more of it.
Great.
You can find out more about Cliodhna O'Connor and her work on our website at scienceforthepeople.ca. We'll be back to learn about neuromyths and the uses and limitations of modern brain imaging techniques right after this.
Science for the People is a weekly radio show exploring everyday life from a scientific perspective. To find out where Science for the People airs near you, or to listen to past episodes, check out our website at scienceforthepeople.ca. You'll also find links to connect with us on Facebook, Twitter, and Google+, or to subscribe to the podcast in iTunes. And now, back to the show.
Welcome back to Science for the People. I'm Rochelle Saunders. I'm here with Dr. Duncan Astle, who works at the Medical Research Council's Cognition and Brain Sciences Unit in Cambridge, and is also a research associate at St. John's College in Cambridge. Duncan leads a program that explores cognition and neuroscience in childhood. He and his team are exploring mechanisms like memory and attention in typically and atypically developing children, attempting to understand how the brain gives rise to such marked differences in cognitive ability, and whether anything can be done to support children with cognitive difficulties. He has previously held posts at the University of Oxford and Royal Holloway, University of London. Welcome to the show, Duncan.
Thank you for having me.
Okay, so we wanted to talk to you today about neuromyths and some of why they exist and why there's all these misunderstandings and confusion about neuroscience. So I think the place to start is actually the definition of one of my new favorite terms, which is neuromyth. What is a neuromyth?
Well, a neuromyth essentially is just a misunderstanding, a kind of commonly held belief about how the brain works that happens not to be true. And there are very many neuromyths. I'm sure you've heard plenty. The obvious one is that we only use X% of our brains, maybe 10%. Another one that you might have heard is that some people are either left-brained or right-brained. So the idea is that the different halves of our brain have kind of different specialisms, and that some people lean more on one than the other. So, for example, if you're left-brained, you might be much more logical and analytical, whereas if you're more right-brained, you're more creative. And I'm sure that some people are more creative and some people are more logical, but that's not really got anything to do with someone being left-brained or right-brained. There's no evidence for that at all, really. We all use both sides of our brain pretty much all the time.
So where do these myths come from? If we know sort of that these are myths, then there's a break in the chain somewhere, right? Where are these coming from?
Sometimes there's a small grain of truth somewhere along the line. So, for example, there are some aspects of our cognition that lean slightly more heavily on, say, the left half or the right half of our brains. For example, language. We know that the left half is heavily involved in language. So there are some subtle differences. And so sometimes these neuromyths have a small grain of truth. But actually, there are loads of neuromyths. And to be honest, it's very hard to see what the grain of truth is. For example, another one is that doing short bursts of specific exercises can alter your brain activity in a very particular way to, say, boost your reading or your mathematics abilities. In that case, there isn't really any much grain of truth in that, so it's very hard to see the kind of scientific rationale for that. One reason, perhaps, is that essentially we all have a brain and we all use it, and this really results in endless speculation as to how it might work and the biological basis for who we are, and this maybe makes us kind of more susceptible to suggestions. And I think sometimes as neuroscientists are really not clear enough. Sometimes we allow things in our community to be published that perhaps ought not to be published. The more exciting the story you can spin around your science, the more likely it is to be published somewhere good. And I think all these things kind of contribute to the kind of perpetuation of some of these myths.
There does often seem to be a little bit of a disconnect between what's actually in the paper or what was actually kind of the experiment that was done versus some of the speculation as to what this means. There seems sometimes to be a really big gap there.
Yeah, absolutely. I mean, I think one reason is that when we as scientists talk to members of the public or to the media, we try really hard to make what we're saying kind of understandable and jargon-free. And I think sometimes we're not really clear enough when we're using an analogy or a metaphor to try and explain what we mean versus when we're actually talking about what we did in scientific terms. And we kind of blur the line between those two things, which I don't think really helps. I guess on one level you could imagine, well, do these neuromyths really do any harm? Does it really matter if we all have lots of kind of misbeliefs about how the brain works? And the more I've thought about it, the more I think that it does matter that we get it right. Because with all these neuromyths around, they're very easily harnessed. And that could be harnessed to sell as products where the benefits are unproved, or encourage us to do things in particular ways. So, for example, I spend a lot of time in schools talking with teachers and children. And in the UK, at least, almost every child knows exactly what kind of learner they are. And they'll tell you that their brain is predisposed to help them learn in a visual way or an auditory way or a kinesthetic way. And that if you try and deliver information that's in a different kind of format, then you'll often be asked, well, how am I to learn this when I'm a visual learner? And you're not presenting it in that format. And actually, the science behind learning styles shows that children are no better in their supposed preferred learning style than in any other. But in this case, it really influences the way teachers try and deliver information, because they think, well, if I don't do something visual, then the visual learners are not getting anything from this. So I think that the perpetuation of neuromyths comes from lots of different kind of routes, but that actually it really does matter that we kind of weed them out, because they can be misused in lots of different ways.
You're listening to Science for the People. I'm here with Duncan Astle from the Medical Research Council's Cognition and Brain Sciences Unit in Cambridge, and we're discussing the ins and outs of neuroscience and neuromyths. It does seem a lot of the times, like your explanation in the different types of learners, that there certainly is a worthy goal there, or a worthy interest in trying to apply neuroscience in a way that helps people, but that sometimes maybe we're taking it a little too far, or we don't really know what we're talking about.
Yeah, in many cases it's extremely worth. I mean, so for instance, the learning styles thing, the aim is to help children make more progress at school, which is one of the things that we're all, in my field, all kind of after. The problem is when you kind of take the step too far. So I think it's really good to encourage teachers to deliver things in different styles. Because actually we know from kind of cognitive science and psychology that when information is delivered in different ways, people are able to retain it more easily. But the problem is when you take it the step too far. So in this case, you take it to the conclusion that children who are visual learners will be able to learn anything that's not delivered in their preferred style. And of course, then that actually starts to hamper the learning, because children have all these kinds of biases as to what they should and what they should not attend to.
So it seems like in neuroscience, there's kind of two main discovery areas. There's kind of the basic sort of really discovery science, how does the brain work? And then there's kind of more, I guess, applied, where we're trying to figure out how we can use what we know in order to do something better, I guess.
So there's an awful lot of science which on the face of it might not seem wildly exciting, in that it's really the backroom kind of stuff. It's those experiments that really enable us to find out, kind of in theoretical terms, how our brains work. So, for example, a lot of the work that we do here is looking at memory and what different types of memory there are, how they're related to one another, what the neural or what the brain basis for those different types of memory is. And most of that stuff is done with healthy volunteers. And really the aim of that kind of science is to discover more about human memory. But then there's a whole separate lot of science, which is really trying to apply that knowledge to practical and health problems. So, for example, you might be exploring the neuroscience of memory in people with Alzheimer's disease. So, for instance, one thing we're interested in is whether we could use brain measures to detect Alzheimer's at its very earliest stages, before behavioral manifestations become apparent. So before the person starts to behave as if they have the disorder, whether you can use neuroscience tools to detect the disorder. And so I think we would argue that these two kind of types of science, the kind of basic discovery science and the kind of translational or applied science, they have to kind of go hand in hand. The benefits that you get for everyday problems and health only really arise when you're also doing the really great basic discovery science.
It seems to me that maybe some of the disconnect when it comes to the public's understanding of neuroscience is that we don't necessarily have a good idea of what the basic or the discovery science is doing, because the translational applied stuff is just so sexy in the headlines. So that's what seems to grab the spotlight.
Yeah, I think that's certainly true. If you can, from our perspective in the kind of neuroscience field, as a researcher, if I could do a study that has a very obvious application to everyday life, then I know that I'm going to be able to get that in a far more high-profile journal. It will have a lot more coverage from the media. Whereas if I do a study that I know is really just about theory, which is very important for our field, but it's unlikely to kind of get the same kind of press attention. So I guess it's really worth bearing in mind that the neuroscience that you see in the public sphere is really just the tip of the iceberg. And neuroscience is a really, really big field, and there's an awful lot of work that goes in behind the scenes.
Okay, so let's shed some light on the day-to-day activities of the neuroscience species and get into the major techniques being used. Let's start with fMRI, which is functional magnetic resonance imaging, correct?
Yes.
So what is that?
So magnetic resonance imaging is probably the kind of technique of choice for most neuroscientists looking at brain imaging. So a way of taking pictures of the brain, so you could have them, you can use just magnetic resonance imaging, MRI, on its own to take structural images of the brain. And so with that you can look at white matter, so that's the kind of thick tracts within the brain. So those are kind of like the highways of your brain. The information can pass very quickly along them. It's very efficient communication. You can also image grey matter, so that's kind of the outer layer, broadly speaking, of the brain. Those kinds of structural images that you get with magnetic resonance imaging are really useful for, say, for example, if you wanted to compare the brains of people with Alzheimer's versus those of the same age who don't have Alzheimer's. Structural magnetic resonance imaging would be ideal for that. And it works essentially using an extremely large magnetic field. And the stronger the magnetic field, the more precise or the finer grained image you'll be able to get. But what you can also use the same type of scanner for is functional MRI. And the way that works is when a small part of your brain becomes active, the metabolic demands of that part of the brain go up. So it needs more nutrients, it needs more oxygen. And the fMRI scanner is sensitive to deoxygenated blood. So when a small part of the brain becomes active, it needs oxygen, so the blood around it becomes deoxygenated, and you can chart that deoxygenation and where it's happening in the brain using fMRI. So those fancy pictures that you see with the colored blobs, the basic picture is done with a structural MRI. The blobs are usually done with fMRI. And the blobs really mean that part of the brain had an oxygen demand, which suggests that it was active.
So what can this tool tell us that we couldn't know before we had it?
One of the real advantages of fMRI is that it's very spatially precise. And that's really useful. Like if you want to kind of chart out the different bits of the memory system. So the memory system actually is really very complicated, and there are lots of different kind of sections of it. And we're increasingly becoming able to distinguish them with these very kind of high spatial resolution scanning techniques with fMRI. But it probably is worth pointing out some of its limits as well. So it might be very good in terms of its spatial resolution, seeing where things happen. It's not so great in terms of its temporal resolution, so seeing when things happen. And the reason for that is because the deoxygenation of the blood can take, 5 to 10 seconds. And 5 to 10 seconds might not sound like a long time, but in brain terms, 5 to 10 seconds, very long time. So it can be quite hard to pinpoint when something is happening. And actually, in neuroscience terms, sometimes the when is a really important question. So if you're doing a behavior where there's lots of different sequences to it, I really want to know when a particular part of the brain is involved so that I can think about what point in the sequence is it really useful for. Then you need the temporal information, and fMRI is pretty poor on that front.
So it sounds like finding the causal link sometimes with fMRI can be really tricky.
It's essentially a correlational technique. If I were to lie you down in my scanner and get you to remember some digits, for example, and then scan your brain whilst you're doing it, it's very hard for me to know that the brain activity that I'm seeing is kind of causing you to remember the digits per se. It's better to think of it as a kind of correlate. So it's a correlate of you remembering the digits.
Can you quickly give us a rundown on the study about the dead Atlantic salmon in the fMRI? Because I think that's brilliant.
So the idea behind this study really was to highlight some of the technical limits. So what we've talked about already are some of the inherent problems of using these techniques. When you actually collect brain scans, you have to take the data through a whole series of steps to go from the raw data all the way to those fancy pictures with colorful blobs on them. And there are various different choices that you have to make as a scientist in terms of how you navigate those steps, the decisions you make about your analysis. And one of the most important steps is what we call controlling for multiple comparisons. So when you do any kind of statistical test, it's taking into account the likelihood of something happening by chance. So, for example, if I was scanning you whilst you're remembering digits, if I were to compare times when you're remembering digits versus times when you're remembering 2, it's about knowing how likely there is to be a difference between those just by chance. But the problem is, when you do a brain scan, you're not just measuring kind of one thing. You're measuring at all points through the brain, which actually is many thousand of points. And of course, that's going to play around with the chances of you finding something. The more times you do the test, let's say I do it 10,000 times in your brain, the chances are some of them are going to be significant because I've done the test 10,000 times at 10,000 different locations. And recently there was a poster at a conference that I was speaking at by Craig Bennett and colleagues, and they did it's essentially a joke, but it makes a really important point. So they had a poster called The Neural Correlates of Interspecies Perspective Taking in the Post-Mortem Atlantic Salmon. And what they did is they took a large salmon that was dead, and they put it in the scanner. And whilst it was in the scanner, they showed pictures of human individuals in social situations that had kind of specific emotional content. And that's a very common experiment that is actually done with human beings. So they delivered these stimuli, these emotional pictures to the salmon, and they then analyzed the data from the dead salmon. And critically, when they were correcting for the multiple comparisons, they used a not particularly stringent way of doing that. And when they did that, they found that they got some nice, significant activations in the jaw of the salmon. So the story is that the jaw of the Atlantic salmon is sensitive to the emotional content of the scenes that the salmon was shown. And of course, that's absolute nonsense. It wasn't. But it really makes the point that when you're doing these kinds of scan, it's really critical that you kind of navigate the technical aspects properly. And it might scare you to know that the threshold that they applied in their study actually is commonly used in many studies that are published in peer-reviewed journals. So that highlights some of the technical challenges of doing this kind of science.
Wow, I love that. And what a great way to make that point.
Really great.
Really inspired.
At the conference, it was absolutely the talk of the conference. Have you seen the poster? Everyone was talking about it, and it created a real buzz. And I think so. It highlighted an extremely important issue.
This is Science for the People. With me is Duncan Astle, neuroscientist and researcher, and today we're looking at the techniques of neuroscience and what they can and can't tell us about our brains. Okay, moving on to EEG, and I'm not going to attempt the pronunciation of what this stands for. I will just embarrass myself. So if you could be so kind and also tell us what it is.
So electroencephalography. Essentially it works by applying electrodes to the outside of the head. You usually put them inside an elasticated cloth cap, and you put that on your subjects, your participants. And inside each electrode is a small hole, and into the hole you force some conductive gel or paste, and then you use a wooden stick or something to position that such that it forms a connective link between the electrode and the person's scalp. What that is able to do then is record electricity from the head, and when different parts of the brain become active, a particular type of cell generates a large enough electrical current, and we measure that current outside the head using EEG. It's a technique actually that I use an awful lot. It's extremely child-friendly. It doesn't sound it, but it is very child-friendly. The downsides are that it's spatially imprecise. So the electrical activity is generated by the brain, but it's kind of smeared across the skull. So it's really hard to pinpoint where in the head is this activity coming from. But the real advantage of it is that it's very temporally precise. So you have almost real-time resolution to know when is the brain, say, differentiating between different stimuli. And that can be really, really useful. So it's not so good at identifying where something is happening, but it's really great at identifying when something is happening.
You mentioned child-friendly. I'm assuming this is because you're not required to be as still as possible while in EEG.
Yeah, so one of the problems with fMRI is that the scanner is actually really noisy. When you go in, you have to have earplugs in, you have to have ear defenders on. It's also very confined, So there's a kind of long, thin bore, and essentially you lie down on a bed, and that is slid into the scanner. So that's not particularly child-friendly in that it's noisy and it's confined. The advantage of the EEG is that it's an awful lot cheaper than fMRI. Secondly, the kids, to some extent, can move around a bit. And the other advantage for us is that you can keep your experiment just as you want it. Whereas when you do an fMRI experiment, you have to take account of the fact that it's a really slow technique, that it takes a long time for this blood oxygenation to happen. And as a result, you have to really slow down your experiment and leave lots of gaps between things, which tends to make experiments very long, which isn't so good for children.
So it sounds like there's a combination you could use potentially in certain tasks of EEG and fMRI to get a better idea of what's happening.
Yeah, so one of the kind of holy grail in neuroscience is the convergence of techniques, is when you kind of combine different techniques. So we're developing ways, for example, of doing EEG inside the scanner. You can imagine that's quite tricky, trying to record electrical fields from the head whilst the person's inside a very large magnet. But with the help of physicists and engineers, we're developing ways that you can combine them both. And so lots of research groups now won't just use one technique. They'll have in their kind of arsenal various different techniques that they try and combine.
Okay, last one. I wanted to talk a little bit about transcranial magnetic stimulation. What is this?
TMS, or transcranial magnetic stimulation. So a real advantage of TMS is that it's what we call an interference technique. For example, if I were to have done your brain scan whilst you're remembering the 7 or the 2 digits, it's really hard for me to know whether the part of the brain that lights up, the part of the brain that's active, is causally involved in your ability to remember the 7 digits. And the advantage of the TMS is that I take your brain scan and then put the TMS device on top of that part of the brain, or position it just above it, and then ask you to do the task again, remembering the 2 or the 7 digits. And whilst you do it, on some trials, some instances, you turn the device on. So you pass a large electrical current through the wires, which turns the coils into electromagnets. That disrupts that part of the brain just for a brief moment. And what I would predict, if that part of the brain is indeed causally involved in your ability to remember 7 things, is that you ought to get worse at doing that when the device is turned on. So it's a really useful way of exploring the actual functional role of different parts of the brain. What kinds of behavior do they cause?
So what are some of the limitations of this new tool?
It's not so spatially precise. It's tricky to know exactly where you're stimulating. Mm-hmm. And then you can imagine that could be a problem. The other problem is that some people have rather thicker skulls. For example, for some people, the part of the brain you want to stimulate is just that bit further away from the coil. And if you don't get the strength of the electrical current correct, then it's very hard to be sure that you're basically delivering the pulse in the right way. So there's lots of technical difficulties with it like that. But it does have that real advantage that it's one of the rare techniques that we have at our disposal that's more than just correlational.
So it strikes me that you guys have a really hard job. Not only is it often difficult to find causation here, but it sounds like a lot of these trials and tests take a really long time, and that to get that instance of replication so that you have, data that you can point to statistically and say, yes, there's something here, it would take forever.
Yeah, it does take a long time. To do it properly takes a long time. A good example, so we've just done a study using brain scanning with 34 children, and the children were split into 2 groups, and they received different types of intervention, and they had a scan before, and they had a scan afterwards. And in between, they had 20 sessions each of this intervention. That study took us about 18 months just to collect the data.
Wow.
Because beforehand, you obviously got to get the whole thing approved ethically. You've got to design the study. Then you've got to recruit the children, which is really tricky to persuade parents to sign their kids up to be involved in a study like that that's going to have such a heavy involvement. Then you've got to get time on the scanner, because of course there's loads of other research groups here who also want to use the scanner, so you have to schedule time. Then you collect the data. Of course, along the way, some of the kids drop out and they have to be replaced, and that child has to start right from the beginning. And then when you get your data, then you have to start the long process of thinking about how to analyze it best. So it's really taken us 18 months to get to the point where we're starting to get some results that we can send to the journal.
Sort of knowing all of this and having this conversation with you, I think really helps understand why sometimes we get these exciting headlines and then not a lot of backstory, because some of the backstory is really complicated.
Yeah. So I think we often try and simplify the kind of process, and actually only a tiny proportion of our work ever really gets kind of public recognition. For example, I mean, this study on brain training that we've done may get some public recognition, but actually there are 4 or 5 studies that have gone before it, which were very important to us, but that will probably never really be read by anyone outside our field. So actually only a tiny proportion of the stuff that we do that really gets into public consciousness.
I guess my question is, do you think that the public could understand more about what you do and could be given more of these details? Or do you think that the way we do it now, where we just kind of talk about the final study that's exciting, is the best we can hope for?
Well, I think that we should credit the public with more ability to understand really what it is involved in science. And for me, the really exciting thing about science isn't just, that this part of the brain does this thing. It's how did we find that out? As in, how did we design the experiment, collect the data, analyze it? How confident are we of the result? For me, that's what makes it all really exciting, is the actual scientific process. And I think that can imagine there are lots of people out there who would find that interesting as well. It's not just interesting what the result is, but how someone figured out how to get to that result. I think that's a really interesting thing. And I would like to see more of that happening, really.
So, final thoughts. As a layperson, keen on science, but perhaps without more than a high school or entry undergraduate level background, how should we read neuroscience headlines? Are there some rules of thumb or red flags we can keep in our brains?
So when you see a story that's, say, about an fMRI study, you've got a picture with some blobs in it, and it's the neural correlates of something very exciting. Remember that some behaviors are really easy to simulate in the scanner. So, we've used the example of me reading you digits to remember. Those kind of memory experiments lend themselves very well to brain scanning, because we can reduce them down to a very simple experiment that is still a lot like the real memory process in the real world. Other things are much more tricky. So the more complicated the behavior you're interested in, the more difficult it is to reduce it down to a simple experiment. So, for example, let's say I wanted to study the neural correlates of moral decision-making. It's really hard to think of an experiment where you get someone to deal with a moral dilemma. And when you get the results, it's really hard to be confident that what you're seeing is the kind of moral part and not all the other stuff that happens when you're faced with a dilemma. So I guess one rule of thumb for those kinds of study is think about what the subjects, what the volunteers actually did in the scanner, and how close or how representative do you think that really is of that process in the real world. So I'd say that's a good rule of thumb. Another type of study you might see is where you see some kind of new intervention or therapy that has some kind of neuroscience explanation or justification. And I think when you see those, you first need to think, is it plausible? Is there a scientific theory behind that which makes sense? Secondly, you need to think, is there evidence for that? And it needs to be more than anecdotal. It needs to be more than hearsay or testimony. Who conducted the study? And are they independent researchers? Did the study focus on the people for whom that interventional therapy is recommended? You need to think, Could there be other explanations for the effect, like a placebo effect, for example? Is there a control group to correct for those kinds of things? And finally, what are the costs of those kinds of interventional therapy? So when you see those kinds of headline studies, the cure for this or the cure for that, think carefully. Is it scientifically plausible? Is there good evidence that's more than just testimony or hearsay? Could there be other explanations that have been controlled for? And what are the costs? How much does it cost relative to the kind of benefit you're likely to get? I think those are pretty good rules of thumb that anyone can apply when they see those kinds of headlines.
Duncan, thank you so much for being here. This has been really interesting.
You're very welcome. Thank you for having me.
If you want to learn more about Duncan Astle or his work, we've provided links to him on our website at scienceforthepeople.ca. While you're there, do click the links to leave a comment on this episode, to connect with us on Twitter, Facebook, or Google+, or to subscribe to the weekly podcast. Thanks for listening, and we'll see you next week on Science for the People.